This is our meeting for Module 3, the third week in this course about molecular techniques and PCR applications. So we'll have an overview and discussion about a very interesting topic from biotechnology, genomics, gene editing. Gradually is going to introduce us to this interesting technology that is called polymerase chain reaction, PCR. Some of you maybe hear about PCR testing during COVID-19. So we'll learn a little bit about some concepts that help to shape up the technology, which would turn out to be the golden standard for measuring gene expression. So in case we want to know if this gene is expressed in certain subject and we are doing certain analysis and we have a treatment and control group, for example, remember the design of experiments, then this is can be one of the techniques or technologies that we can go and apply and see if that is confirming that this gene was expressed or not, for example. At the end, we'll have also a discussion on graphical abstracts, something that is a part of some publications, and we'll see how it's something we can take advantage of to learn more about the work. So as I mentioned, it's more about PCR. Another level, as now we move from our background with molecular biology and genetics, it's now the beginning of learning more about technologies. So today we'll speak about PCR. And related concepts as well, like fluorescence techniques, and what's the origin of that, and some interesting applications. So as we speak about this, the point that we need to keep in mind is that in terms of diving into certain findings in the space of life sciences, looking into this PCR protocol can expose us to one of the important techniques in genetic analysis. For example, molecular labeling and tagging and fluorescence concepts that we'll kind of skim through in this part of the course helps in finding interesting way to measure a gene expression and target specific genes. And this kind of technologies or concepts have, in fact, these concepts have helped to become part of several applications or technologies, including, for example, CRISPR technology, which is now widely used for gene editing, for example, and as well as adapting PCR, for example, for single cell analysis. And this is all cutting edge research indeed. This is what we want to dive into today. Yet, before that, let's see how you are doing so far with the course. So right from the beginning until now, we're kind of visiting, yes, lots of biology concepts and molecular biology and lots of terms. For some, it is a kind of review given their background. For some, it is a dense list of terms. And it can be sometimes difficult to get the full picture right from the beginning. And therefore, it is a good time to think about the best way to manage your own schedule, look into the reading material, watch the recordings. Personally, as I discussed in the office hour, I love to go and find if there's any, on my free time or so, if there any YouTube or animation or something that explained, for example, CRISPR technology? Is there any reference? And sometimes I like to interact with generative AI models, back and forth with lists of questions, try to build my understanding or read some reference material. But again, it is a learning process. It's going to take time. And gradually, you will see yourself diving into more and more computational and programming things. So part of the revision that I want to do here at this time, first, I want to refer to what Nikhila had shared with me, and I think it is very interesting. It is related to a snippet from a book called Code Breaker that talked about that Francis Crick. And if you remember from our discussion before, Francis Crick is this big name that contributed to finding the double helix structure, the helical structure of the DNA. And that Crick talked about, regretted calling the central dogma a dogma, using the term dogma, because dogma was something that shouldn't be questioned. And the reason I mention this here, if there's anything I would like to review with you and ask about, is certainly certain central dogma of biology. But it is such an interesting hint that, that it can be questioned, and science is all about raising questions and diving more into information. So thanks, Nikhila, for sharing that. I found it very interesting to share with all the students. So this is one thing, as well as I want to go over some questions, part of review, and then part of you doing the treasure hunt exercise that we discussed earlier. So one of the questions would be that to think about how did DNA replication, how does DNA replication work? And we discussed the most beautiful experiment in biology. Here's one question for your treasure hunt exercise. What are histones and what they do? Okay, I want to refresh your memories with some of these discussions. And what are histones? What do they do? Also, we discussed different ideas about the directionality. I want to confirm that you understand the directionality. So what is 5' and 3'? Where do they come from? This is the second question related to our treasure hunt weekly activity and the bonus marks related to it. Okay, so what is 5', what 3', and why that matters? Can you give me an example related to replication or so? Which is going to bring me to another question. Why do we have Okazaki fragments on the lagging strand? And again, this is the third treasure hunt question. Why do we have Okazaki fragments on the lagging strand? So 3 questions so far. I think that is enough for the treasure hunt. We also discussed so far differences in prokaryotic and eukaryotic translation process. We looked into the mRNA and different types like polycistronic mRNA, which when we give the example of FLAC, the protein, operin, and that how it encodes for several proteins in prokaryotes. We looked into concepts like telomerase, 3', and so on and so forth. So many things that we have discussed, differences between prokaryotes and eukaryotes, and also some of the things that we have looked into. Promoters, okay, what is a promoter? Transcription factor, and so on and so forth. The Tata box, remember what is a Tata box? So it is, it is part of the, I would say, hidden message telling the transcription factor in the promoter area to bind here. It is encoding this message. So once it binds there, it initiates the transcription process. So remember, central dogma is all about translation, transcription, replication, translation, transcription. So initiating the transcription is very important for the production of proteins. We went also, we skimmed through some concepts like exons and introns and how they play a role. So exons are the coding sequences of the gene that are expressed and translated into proteins. Introns can play a role in regulating this gene expression. We looked into poly-A for an RNA and so on and so forth. I agree, these are a list of, some biology and molecular biology terms, more to come today. I would perceive it from the angle that there are lots of biological processes that are happening. We need to understand that we have to be careful when we are diving into a bioinformatics project, particularly about the experiment design and what is our aim and hypothesis, because there could be many things coming together. And also to know at the same time that some projects are really complex, which would require good collaboration. So generally in bioinformatics, if you want to put a touch on a certain problem, or leave an impact, you are looking into coming and working with the team and trying to sort out different angles of that project. So I hope this helps. Now, I've listed 3 questions as kind of treasure hunt exercise. Please answer those and share those with me along other questions that may come throughout the lecture. Also, the other questions, I hope they kind of motivating your memory. If you were not sure what is this question about, again, go and find out or reach out to me and I'll try to help you to find the right answer. So now we are going to move into more discussion, which is called, the topic here is about reporter gene fusions and fluorescence. We want to break down again some of these terms and, try to see what is reporter gene and fluorescence. And the way I would like to use this is start actually using this green color for fluorescence. We know fluorescence is about this interesting light pattern that we can sometimes see with toy balls. So some toy balls that are shining in the dark light after you expose them to the natural light or some blue LED light or some type of light, and then they absorb the energy coming from the source light, and then they are going to release some light. Once we turn the lights off, we see, oh, this is a glowing object. So this is glowing object is what relates to the idea of fluorescence. And nowadays, maybe one of the interesting trademarks, as myself like this hobby of having water tank with fish inside that. So maybe some of you heard about the glow fish. It is this type of fish that can glow, okay, and give interesting lights when you have the blue light on top of your fish tank, okay. Anyway, so a reporter gene, it is an exogenous coding region. It is exogenous means it's not part of our system. It's coming outside through some mechanism with some promoter sequence. And we know now promoter is this area that is going to give the initiation of transcription. So when we design something and we try to inject it somehow into the biological target, we are looking into, of course, to be able to measure a certain amount, because this process may lead to the production of a protein or enzyme that can be easily detected and measured. So there are so much into this. We're trying to dive into this. The idea, to make it simple, there is a certain technology that's going to take advantage of this toy ball that we have and release this light under certain conditions, that would help me to detect if there is some expression of that gene or not. So fluorescence in this case is provide us a powerful tool. You can see these pictures here all about biological subjects and cells and different types of targets where we see fluorescence slides. Very amazing figures that we can watch into. In fact, the actual laboratory experiments can be sometimes fun to do. And fluorescence is going to be, and again, this kind of chemical reaction, this slides that is coming in a way that is very interesting and appealing to us. This slide would turn out to be very powerful, a very powerful tool for us to measure certain information. It is going to be part of several technologies. We will look into some today, even when we're going to talk later on about gene expression technologies, such as microarray and the different lasers that are used in that technology and how they scan the chips, so on and so forth, to see the release of that light. So the release of this light is really helpful into exploring and discovering the inner working of cells and the products. Fluorescence facilitates understanding several types of cellular processes. For example, what's called molecular specificity. So fluorescence probes or tags can be attached to proteins. These fluorescence tags that would help to release the light, okay, can be attached to proteins, nucleic acid, or other biomolecules. This would help in tracking location, okay? Other types of applications, subcellular localization. So by tagging a protein or other molecules with fluorescence markers, it can be possible to determine the precise location with the cell, maybe in the nucleus, mitochondria, and so on and so forth. Quantification, and this is something I want to write down here, because it's key to the discussion today and other things to come. Quantification as one of the good applications, of fluorescence. The intensity of fluorescent signal can be quantified. It is light that we can scan. And imagine again, sometimes you have your toy ball. I hope everyone had this nice experience, these glowing objects that we can have sometimes. Sometimes you see them very strong. It depends on the type of the object and so on and so forth. But sometimes you're able to see, oh, it is such a strong color. That is something that is measurable and quantifiable. This would allow us to know the concentration of a particular molecule, for example. Another interesting application for this fluorescence technology is what is called multiplexing. Multiplexing. And it's when different fluorescence probes emit light at different wavelength. We're going to come to this and see what is wavelength. Okay, it's a way to measure the types of the different waves. And remember, we have different electromagnetic waves, and there is a range on the spectrum of these electromagnetic waves, things that are visible to our eyes and things that are not visible. And yet each have, as a signal, has a wavelength. So this multiplexing allows researchers to tag multiple cellular components and observe them in parallel at the same time. There are more applications for fluorescence technologies. It gives and provides, of course, a direct kind of way to observe what's into the cell. Okay, and as we are looking here, this is very interesting story as we try to dive into what you see here is the jellyfish. Okay, this is the jellyfish. This was the source of some interesting idea. a scientist that got interested with this type of fish in the early 1960s called Osamu Shimomura, a Japanese chemist, and was intrigued by the bioluminescence of this jellyfish that's called fluorescent jellyfish, or Aquaria victoria, okay, as the scientific name. So Osamu was very interested. What is unique about this fish, right? He was able to see this green fluorescence. What you see here is what Osamu observed. So he began studying this jellyfish to understand the source of its glow. Why it is glowing? And he was intrigued. Is it something to do with the protein? What is happening there? So through the research that Osamu Shimomura did, he realized that there were two proteins responsible for the jellyfish activity in terms of bioluminescence. Okay, the light that comes out of it. So there is what's called. Let me write this down. Oh, well, I'm going to use the color related to this first, the first protein. Okay, so this is equiron. Okay, this is the first protein. Okay, that Shimomura explored. So this protein emits blue light when it binds with calcium ions. Okay, so this is a source of blue light. And another very interesting protein, which is going to bring lots of discovery and turn out to be an amazing breakthrough, is GFP. Okay, GFP, or green fluorescent protein. GFP for short, it is green fluorescent protein. And this protein actually absorbs the blue light. Remember the example of our toy ball, okay, in the dark room. This protein is able to absorb the blue light emitted by equiron, the first protein, okay, and then re-emits it as a green light. So there are two types of light. One that is absorbing and one that is going to be emitted. Okay, it's going to absorb the blue light, and actually it's better to change the color of the light that's going to absorb to be more accurate here. Okay, absorb. Okay, and here this is going to be the light that is emitted. Okay, or let's write emission. Okay, so it's going to re-emit green light. That makes the jellyfish and gives it this characteristics. Okay, so anyway, it was very interesting research. Shimomura and his team tried to purify this protein and isolate that as a separate protein and describe its properties, and then that was really groundbreaking because of the potential. But this ought to be that it can be used as a biological marker, as a biomarker. And this gene, the gene encoding the GFB protein, scientists then realized that let me change the color here again, not to confuse you with the lights. And this lecture is about lights, and especially the green light, okay? Especially the green light here. Bear with me. So the gene that is encoding GFP protein, the green fluorescent protein, they discovered that could be inserted into the DNA of other organisms. Okay, DNA of other organisms. Okay. Now here is a question for you, and this is question 4. And usually I don't give the number of questions for the treasure hunt, But again, because I gave 3 in the beginning among lots of questions, so at least you remember there were 3 questions before, since we had a review today. So here's a question. If we know that there's a gene responsible for a green fluorescent protein, and that gene can be used and inserted into DNA of other organisms, okay? And another piece of information, when it's inserted into other DNA of other organisms, it can cause them to produce the fluorescent protein, okay? So here are 2 things. Imagine that this gene can be put in one cell, and that cell eventually is gonna transcribe, go through the process, and then we will have what? What are we gonna have? Again, a release of fluorescent light. The question is how this can be helpful for researchers or for you as a scientist. Certainly something that would relate to visual observations. Can you give me any application that you can think of how we can take advantage of such idea? And again, let me remind you, you don't need to give the correct answer. You don't need to go and look online. Any answer is acceptable and fine, but just I want to see what comes to your mind when we are discussing this. Okay. This is the story of jellyfish. Now, as you know, we looked into the green fluorescent proteins, but they are not just green. Okay, there are other types of derived proteins. For example, there is what's called the monomeric red fluorescent protein. This one. Okay, and monomeric means it is on the similar range and levels of colors. So you can see here we go all the way from dark red into very light red in this area. So there are other types of molecules that can be motivated to exert and emit different types of lights. Now, one would come and ask, why do we care about these different examples? Is it something just to be a piece of art, or how do we care about this? It's a very good question, okay, as to see these colors and see that such kind of colors can be generated by certain mutations that we can apply to the GFP and eventually have different colors like this. And that's why when you go to pet store, you want to go, and I'm not promoting the glowfish actually, okay, but again, just to try to link it to your thinking, but you may go to the section, see this glowfish, or you can look online at glowfish and see how they have different colors. But here is the discussion is about a technology that we can use for certain applications, and gradually we're going to dive into some of these interesting applications. Yet the question is, why do we care about different lights in this case? Remember when I told you that there's excitation or kind of absorption and then emission. So there is a source light. When we talk about light, we need a source to give it is called the illumination source. It is the source that is going to give the light that will hit a certain object, okay? Think about this like light bulb you have in your room, or the sun in the sky, or any other illumination source. And once it hits this object, okay, some light is going to be reflected, some is going to be absorbed, or maybe everything is reflected. It depends on the type of the object. Some molecules, however, they are going to take that and start absorbing light and then start emitting their own light. So therefore, we have two levels of light that we're exploring here, which is the excitation and emission. Now, without necessarily the need to dive into so much of details here that may relate to a little bit more chemistry, I believe, and physics, one of the observations that we can look into here is that this is 600 nanometers, okay? And what do we find here? There is, so this wave, I'm trying to check, it's Mach cardinal color, so this is the one. So we have one wave here, okay, of that we are capturing here, and there is MK, okay, this one, okay, with only a small difference in the emitted excited level of light. So the light that we expose these molecules to, which is an energy that is we are paying for. So with just a small difference, we're talking probably here about, I think, 15 to 20 nanometers, and the details are there in the reference paper anyway, yet you gain a high range of excitation with the Mach cardinal. In other words, when we are looking into designing certain experiment, it depends. Maybe we want to look more into the wavelength and the energy needed to be able to decide on which fluorescence we want or we are interested to work with. Okay. Mach cardinal in this case as a color have excitation that peaks by just a difference of 15 nanometers. Okay. And Mach cardinals would have a broader spectrum, resulting in more absorption, okay, of light. So anyway, it is just to help and tell you that there are different ranges, and depending on the type of experiments, actually, this is not only green, and we can come up with another thing that we can work with. So here is certainly one question that we want to ask after talking and having this discussion so far about fluorescence of gene. How can we take advantage for that? And let me relate this to the question that I've asked you shortly back. Okay, what can we do? What can we do with that? So what I want to point here on the slide is that this can allow us for direct qualitative and quantitative measures of certain biological activities. Okay, so what are we looking into is, let me revisit let's make it green and then talk a little bit about, sorry, bear with me, GFV, G, green fluorescence protein. Okay, this protein that can be inserted into the DNA of an organism can cause the organism to produce the GF protein. Okay, eventually that's what makes the subject or the target show a glowing green light. Something I mentioned that can be used to tag certain genes, this kind of strategy. Okay, so what I want to explain here is a process where there are certain steps. Let me try to draw this for you. The process of application, and the application here that we're going to look into is try to measure intensity of green fluorescence and see how that can relate to certain experiment. Okay, so first of all, as a scientist, I start by identify, this is, I would say, you can think of it as the first step, identify gene of interest. Okay, identify a gene of interest. Start by selecting specific gene you want to actually study. Okay, a specific gene that you want to study. And as you decide that specific gene that you want to study, you want to see if that gene in that target cell is maybe transcribed with large quantities or not. Remember, this has to do with transcription, mRNA messages that comes out, and we can measure mRNA and so on and so forth we want to quantify that. We want to know the technology that can be used to quantify that. So second step, after we identify gene of interest, the second step here is to clone a gene into plasmid. And so here I have some interesting terms. Once we know that this is an interesting gene, any type of gene, we want to put it on some DNA structure. Usually that's called a plasmid, okay? We are working with what's called a plasmid. Plasmid itself is a small circular piece of DNA, usually found in bacteria, okay? It is separate from the chromosomal DNA found in cells and bacteria. So we can get some kind of DNA, and then we can have and clone the gene of interest into that specific gene, okay? So we can insert or we can find that gene and then try to have it inside a cell. Now, something as we work with these, what's called plasmid cells, again, we have identified gene of interest, okay? We have identified the gene of interest. Now we are going to work with in cloning the gene into plasmid. Third step here would be insert, let's look into this, insert GFP gene, and here what I want you to pay attention for, GFP gene, okay, next to gene of interest. Maybe this line should have been the green one, GFP, the greenish one. Everything else just a normal text because this is the green fluorescence protein. So what are we saying here? One step, we identify gene of interest. Second step, we say we have to have a cell where we'll put this gene, okay? And we may need to clone this into some type of cell as a protocol, as a setup, because we don't want to, we want to have this targeted and simplified somehow. Anyway, next we need to insert the green fluorescent protein next to it, right beside the gene of interest. This is how we do it in the plasmid. Okay, so now you can think of having two genes next to each other. That's what's going to take us to the fourth step, which is produce fusion protein. Okay, and produce fusion protein. It is, now we worked with the genes. However, the cell's machinery is going to read the genes and create a combined protein of both. Which genes? Gene of interest and GFP. Gene of interest and GFP. Okay, so this is as we get into the fourth step. And then we had worked with the DNA, the copy of, we looked into the plasmid, right? That is a circular piece of DNA. Now what we want to do, as we have now the fused protein, we want, as part of the plasmid, we want to put the plasmid, okay, put the plasmid into, introduce plasmid into cells. Introduce plasmid into cells. Now, I am sorry, I'm just writing text here. It's very difficult for me to go and sketch all of these details, but I'm going to show you some figure next on the slide that hopefully this would make sense as you review this material. Sometimes it might be useful to go and repeat the recording, okay? So introduce plasmid into cells, okay, that piece of DNA. So actually just insert the plasmid with the genes into cells. That is, in summary, what is going to be done. And then next, cells are going to produce the fusion protein themselves. Cells will produce fusion protein. Okay, that is a next step. So we went from producing fusion proteins in terms of the design at that level of having 2 genes, and now it's going to be a cell production. What is going to happen next is very interesting. As this protein is going to be released, this fusion protein, we're going to observe. Okay, observe. What are we observing? Okay, under some fluorescent microscope or any type of technology, what are we going to observe? What? That is a question. Okay, what do you think we are observing? Okay, think about it for a moment and please share with me the answer as part of our treasure hunt. Okay, we are observing what? Intensity of green fluorescence. Okay, of green fluorescence. Good. Now, so there is some way of how we take advantage of, I mean, if you get nothing from this slide, but that there is a protocol into how we can take the GF protein and join it with another gene. This is going to get me into this picture here, that there are different ways into how we can do this. You can see there's example A, B, and C. The place where you can put the GFP protein or gene to help in releasing a light when certain process is going to take an action, there can be several examples to this, as we see in this figure. First of all, there is what's called transcriptional. Transcriptional. Transcriptional reporters. Okay. Transcriptional reporters, I'm starting with the figure on the right. It consists of promoter fragment from a gene of interest driving GFP. So the promoter fragments is few kilobases immediately upstream of the start codon. So this is where the promoter directly comes the GFP protein. This is what we call transcriptional reporter. There is what's called, and it's actually provide, again, it's going to be part of a tentative expression pattern. There are what's called another type that we call translational reporters. And in this case, the fusion of the GFP and gene of interest happens as you can see. A translational reporter includes the entire genomic locus. GFP can be inserted here at any point. Okay, at any point. In this case, what is different between translational and transcriptional? Translational have more information like exons, introns, 3' UTR. So it is more accurate in terms of information as we fuse these 2 genes. And finally, there is another 1/3 type, it's called SMG1 based reporter, which are transcriptional reporters that aim to include some cis-regulatory information. So anyway, GFP in this case, including stop codon, is inserted between the promoter and the first exon. Okay, between the promoter and the first exon. So in summary, in a nutshell, we can actually have different ways into how we can fuse this gene into the different places or areas of interest, eventually helping us. And what is the aim of all of this? What is the aim of having these fusion genes? Is to look into the production of fusion proteins. As we put them into cells later on, we are inside the cell. The fused gene is expressed as a single fusion protein. And this fluorescence, since it's part of it, is going to help me into quantifying expression levels. Lots of interaction, lots of dynamics are going to happen, but this is the idea that we want to capture as part of understanding for this. Okay, and the figure here on the left that we're looking into is referring to interesting technology. Okay, so there are several terms here, not going to dive so much into this, but there is what's called one key idea here as adenine base editor. Adenine base editor is kind of genomic editing technology. And this is going to bring us insights about CRISPR, what is called CRISPR, which also try to use this whole idea of having some kind of gene inside a plasmid or kind of cell that we can send into the subject and then can play some role in editing certain areas of the gene. And ABE here, or adenine base editor, can directly convert AT base pairs into GC that we see here. Okay, can do this kind of manipulation or of changes or editing into the target cell without introducing double strands. Okay, so in this paper they talk about trying to dive into this protocol and how it can they develop this Beyond base strategy, which stands for base editing on to quantify ABE efficiency. The original adenine base editor techniques for genome editing and how fluorescence eventually helped in measuring that kind of information. And there are very interesting terms here like gRNA target, and it was such an amazing discovery as well on its own, which is very important. It's called the guide RNA. It is something that can be designed to help as well in the editing process. There's what's called ATG removed GFP, and it's another as well interesting term that I would suggest if you are interested to look into. Okay, this term. What I want to add here is, yes, ATG removed GFP refers to the version of GFP in which the start codon, that is usually called ATG, has been removed. Okay, this is usually crucial to initiate the expression of the sequence, and so getting translating mRNA into protein. If that is removed, by removing the start codon, the translation of GFP then would be prevented. Okay, would be prevented. And so I'm going to stop here, and then we're going to continue in the next part. Thank you so much.